Let us discuss about respiratory quotient, RQ. Here in the respiration, oxygen is consumed and carbon dioxide is released. So in aerobic respiration, here the ratio of CO2 evolved, here carbon dioxide evolved and oxygen is consumed. In aerobic respiration. The ratio between the CO2 evolved to the oxygen consumed in aerobic respiration. So we can write like this, RQ values. So the volume of CO2 evolved during aerobic respiration by the volume of oxygen consumed. So in aerobic respiration, different respiratory substrates are involving. Here glucose directly it is participating in the aerobic respiration from the starting point. But fats and proteins they are not directly involving. So first they are converting into different components, then they are entering into the aerobic respiration. So different respiratory substrates are involving. Here the respiratory substrate is if it is carbohydrate, the respiratory substrate is carbohydrate. So we know the aerobic respiration equation here, C6H12O6 like 6O2, 6 CO2s are released. So here if you observe the carbohydrate or glucose, mainly here in the glucose, the carbon ratio, here the ratio or the number of carbons and oxygen same. So here only 6 oxygen molecules are enough to oxidize this glucose. Here if you observe the RQ of carbohydrates, the 6 CO2s are evolved and 6 oxygen molecules are consumed. So the same number of carbon dioxides are evolved and same number of oxygen molecules are consumed. So the ratio is 1 for carbohydrates. If the carbohydrates are involving in the aerobic respiration, the RQ value is 1. Why? Because here the ratio between the carbon and oxygen it is same. So number of carbons and oxygens, only 6 oxygen molecules are enough to oxidize this component. So 6 carbon dioxides are released, 6 oxygen molecules are consumed. The ratio is 1. If the fats are involving in the aerobic respiration, the RQ values are always less than 1. Here if you observe one fat, a tripometin, C51 H98 O6 taken twice. That is the formula of tripometin. If the tripometin, it is a fat, if it is involving in the aerobic respiration, here 145 oxygen molecules are required. Oxygen molecules are 145. Only here 1 or 2 CO2s are released. 98 water molecules and some energy. Here in tripometin, if you observe the structure, the formula, here the oxygen ratio, here the proportion of oxygen to carbon, here it is oxygen proportion is very less. So oxygen number is very less, carbon number is more. So for the oxidation of this fat or tripometin, it requires more oxygen. In the structurally, oxygen number is very less, carbon number is more, carbon and hydrogen number is more comparatively oxygen. So it requires more energy here. It requires more oxygen molecules. So 145 oxygen molecules are required. And here 1 or 2 CO2s are released. So if we write here 1 or 2 CO2s are released or evolved. The volume of CO2s evolved. So 1 or 2 CO2s are evolved and 145 oxygen molecules are consumed. So the ratio is 0.7 approximately. Here 0.7. So always the fats, if the fats are involving in the aerobic respiration, the RQ values are less than 1 only. So that is 0.7 for tripometin. Next, if proteins are involving in the aerobic respiration, in the proteins also, proteins are involving in the aerobic respiration. So the in protein structurally, here it is, proteins are also having very less number of oxygens. So like fats, in the fats oxygen number is less comparatively carbon and hydrogen number. So they require more oxygen. Here proteins are also same. Here the oxygen number is very less comparatively carbon and hydrogen. So RQ values for proteins always less than 1. So approximately 0.9. For fats 0.7, for proteins 0.9. But both are here less than 1 only. Less than 1. The RQ values are less than 1. And organic acids. In some special conditions or specific conditions, the organic acids also involving in the respiration. So if the organic acids are involving in the respiration, the RQ values are more than 1, greater than 1. Sometimes 1.33, 2, 3, 4, like this. More than 1 only the RQ values are present for organic acids. So why here the organic acids are having more RQ values? Here in organic acids, the carbon number is less, oxygen number is more. So the proportion of oxygen to carbon and hydrogen, it is high, or more number of oxygens are present in the organic acid. So these require very less oxygen. So these require less oxygen molecules. Why? Because here the carbon and hydrogen number is here more. So I am repeating, organic acids. If organic acids are participating in the aerobic respiration, the RQ values are greater than 1. Why? Because here the organic acids contain here more oxygen. Here structurally the oxygen is more comparatively carbon and hydrogen. So here more oxygen is present in the structurally. That's why they require very less oxygen for oxidation, for respiration. So here if very less oxygen is consumed, the RQ values are here more. So carbon dioxides are more, carbon dioxides are released, but very less oxygen is consumed. So the RQ values are here more than 1. Next here anaerobic respiration. Anaerobic respiration. In anaerobic respiration, oxygen is not consumed. So if you observe the anaerobic respiration, here C6H12O6, no oxygen is consumed. Here ethanol and carbon dioxide, 2 CO2s are released. Here 2 CO2s are released but no oxygen is consumed. So the RQ values are here 2 CO2s are evolved but 0 oxygen molecules are consumed. So 2 by 0 it is infinitive. So for aerobic respiration the RQ values are infinitive. So here carbohydrates, fats, proteins, organic acids, so these are respiratory substrates. If different respiratory substrates are involving in the respiration, the RQ values are also different. So how can we measure the respiratory quotient values? Here respirometer, by using a respirometer, here in controlled conditions these respiratory quotient values are measured. So how much oxygen consumed in the aerobic respiration, how much carbon dioxide is released. So depending on the consumption and release of carbon dioxide, the RQ values are depending. And carbohydrates, fats, proteins, organic acids, they are having different structures, and the carbon and oxygen ratio or the proportion of oxygen carbon, that is different. That's why the RQ values are also different. If the oxygen is more, so very less oxygen is consumed. Structurally if oxygen is more, very less oxygen is consumed. If structurally oxygen is less, more oxygen is consumed. That's why the RQ values are also different. So it is about respiratory quotient. But here anaerobic respiration, in the anaerobic respiration the RQ values are here infinitive. Why? Because here no oxygen is consumed. Now all these oxygen is consumed. That's why here it is more or less than 1 or equal to 1. But here no oxygen is consumed in anaerobic respiration or for fermentation also. In fermentation also no oxygen is consumed, but CO2s are released. So here 2 by 0 it is equal to infinitive. So for anaerobic respiration the RQ values are infinitive. So these are the respiratory substrates in aerobic respirations, and anaerobic respiration RQ values are infinitive. It is about respiratory quotient.